Dear student, I am Dr. Prakash Mungli, professor in Biochemistry. So in this video, I will be explaining you all the factors that affect enzyme activity. So there are three important factors that can affect enzyme activity. One is substrate concentration, other is temperature, and the third is pH. Now there are some other factors that can also affect enzyme activity, and that is enzyme concentration itself, product concentration, presence of inhibitors or activators of enzyme, and also availability of coenzymes. Now let's get into what is the effect of substrate concentration on enzyme activity. Now in order to understand the effect of substrate concentration on enzyme activity, we really must consider that enzymes remains constant at all times in our cells, because there is a range for enzyme in our cells. Like when you compare the concentration of enzyme, any particular enzyme that is present in my cells compared to the concentration of enzymes that is present in your cells, so it always remains constant. There is a normal range for every enzyme and it doesn't alter. If there is, say, example, alanine transaminase enzyme. So the normal range for alanine transaminase is 20 to 40 international unit, and this 20 to 40 international unit doesn't change under any circumstances unless if there is any disorder. Like, so whether we measure alanine transaminase in one person or other person, it should be in the range of 20 to 40 international unit. If there is less than 20 international unit, that's the deficiency of enzyme. If it is more than 40 international unit, it is an excess of an enzyme. These are abnormalities. So under normal conditions, if a person is normal, any enzyme that you take an example, it remains constant. That's why if you look at this particular figure here, so only 6 enzymes are mentioned. The red colored ones that you are seeing in the figure, those are the enzymes, and there are 6 enzymes in 3 categories that is shown in the figure. So 6 enzymes, and now look at the x-axis in the plot. X-axis indicates substrate concentration, and the substrate concentration is mentioned as moles per liter, and it can change. It can be millimoles per liter, it can be micromoles per liter, depending on what is the substrate concentration that we are using. And in the y-axis, we have initial velocity of the reaction mentioned, and the initial velocity of the reaction is the units for that is moles per liter per second. Now basically here in this particular graphical representation, product formation is mentioned with the red line. So the red line that is there in the center, it is the product formation. Now let's see in the initial substrate concentration how an enzyme will react. Now in the initial substrate concentration, we have 6 enzymes but only 2 substrates. That's the low substrate concentration. We have plenty of enzymes, and there are 6 active sites at least in the figure here. Every enzyme has at least 1 active site, and the substrates are only 2, active sites are plenty, and these substrates, they have plenty of active sites to go and bind. So substrate will bind, product will form, and there will be slow initial rise in the product formation, and the rise in the product formation, it is proportional to the substrate concentration here. As you see, as the substrate concentration increases, product formation will also increase. It is kind of a straight line there. In the initial substrate concentration, so product formation remains, means it is directly proportional to the substrate concentration. Now, this type of reaction, enzyme catalyzed reaction, is referred as first order kinetics. So in the first order kinetics, so velocity of the reaction is directly proportional to the substrate concentration, because there are plenty of active sites are available and the substrate concentration is less. Now let's look at the number 2 in the group number 2 here. Group number 2, as you see, there are 6 enzymes which is constant, and now there are 7 substrate now. because we are constantly increasing the substrate concentration going from left to right. So as we are gradually increasing the substrate concentration, now there are 7 substrates and only 6 enzymes are there. All the active sites of the enzymes are occupied. That means product formation is kind of now getting into saturation. And the one of the substrate that is waiting here, not getting a chance to bind to active site, it's going to get the chance to bind to an active site only when the substrate that is already bound to an enzyme is released as a product. Only when that product is released, then only the substrate which is waiting can go and bind to an active site and can become a product. So until that time it has to wait. So that means enzymes are now getting saturated. So that's why you see the slight deflection in the reaction velocity curve, that is the product formation. It is kind of getting deflected, going into a plateau stage. Now let's move on to third category there, third group. In the third group, as you see, again there are 6 enzymes because enzyme remains constant in our body, in our cells. Now we are constantly increasing the substrate, we are going on adding substrate. Now there are plenty of substrate, much more than the enzymes that are available. So all the enzymes active sites are full. That means there are plenty of substrates are still waiting. So only when substrate that is already bound to an enzyme's active site, only when it is released as product, other substrates can go and bind. So at this point in time, only 6 substrates can bind, 6 product will be released, and 6 more substrate will go and bind, 6 more products will be released. That means at any given point in time, only 6 products can be made here, and that remains constant. And that's when a reaction velocity curve, it becomes plateaued, and that's the plateau state there. So that's the maximum velocity of this particular group of enzyme, and that maximum velocity, that point is taken and it is interpolated to the y-axis, and wherever it meets on the y-axis, and that point we refer it as Vmax, and that is the maximum velocity of the reaction. And the Vmax value, it is still moles per liter per second. So that means in an enzyme catalyzed reaction in our body, always we have a constant amount of enzyme. Only thing that changes is the substrate that we are providing to these enzymes. Okay, and for our the purpose of understanding, in our body we will have plenty of enzymes. We will have more than sufficient concentration of enzymes in our cells. Only thing is these enzymes will be waiting for the substrate, and that means since there are plenty of active sites available, less substrate is there, majority of these enzymes will be working at first order kinetics, that is velocity of the reaction is directly proportional to the substrate concentration. So that's where we are most of our enzymes. Some of our enzymes, they will be saturated with the substrate. There will be the concentration of these enzymes will be much lower than the substrate that is available. Majority of time they will be saturated with the substrate, and they will be working at 0 order kinetics. That is the number 3 here in this particular figure is the saturation kinetics, which is also called as 0 order kinetics. And in 0 order kinetics, the velocity of the reaction is independent of the substrate concentration, because you cannot increase the velocity of the reaction unless we increase the enzyme concentration itself, because there are plenty of substrates, only limited enzymes are available. Unless we change the enzyme concentration, we cannot change the product formation, because it is remaining constant, because enzymes are already saturated. Okay, and that is the saturation kinetics here. So most of our body enzymes, they work at first order kinetic, group number 1 in the figure. That means we have plenty of enzymes, less substrate. So enzymes are available plenty, product formation is dependent on the substrate that is available. So reaction velocity is directly proportional there. So very few enzymes in our body, they work at 0 order kinetics, because there will be plenty of substrate and less enzymes available. So they are always saturated, they are working at Vmax, or the maximum capacity. So this is how we can extrapolate the information that is there in this particular graph to what actually goes on in our body. This is what is the understanding that you should have. Okay, so the understanding here is majority of our enzymes, they work at first order kinetics, because we have plenty of enzymes already available. We are waiting for the substrate. Whenever substrate is given and that substrate is immediately converted into product, velocity of the reaction is directly proportional to the substrate concentration. Very few of our enzymes in our cells, they work at zero order kinetics, where enzyme concentration is less, or they have low Vmax, and basically they have plenty of substrates waiting, and so the velocity of the reaction is independent of substrate concentration for them, because even if you go on increasing the substrate concentration, you cannot change the velocity of the reaction when the enzyme has got saturated, right? Because in this figure, only 6 products can be formed at any given point in time. Even if you increase the substrate concentration, you cannot change the velocity unless you change the enzyme concentration. Unless you add more enzymes, then you can increase the velocity of the reaction. Unless a cell synthesizes more enzymes, unless the cell increases the concentration of enzyme, so product formation cannot be changed whenever enzyme reaches saturation. So this is what is the effect of substrate concentration on enzyme activity, and this particular model that we are studying now, it is explained by Michaelis and Menten scientists. These are the two scientists that who have explained this particular enzyme kinetics, which is referred as Michaelis-Menten model for enzyme kinetics. It is an equation relating reaction velocity to the substrate concentration, where enzyme binding with the substrate, making enzyme substrate complex, and enzyme is coming out of the reaction unaltered and the product is released. So there are a couple of parameters here. One is the substrate concentration, other is the initial velocity of the reaction, and the third is Vmax, maximum velocity of the reaction. Now looking at these parameters from the previous slide, now we have substrate concentration, which is in millimoles per liter. We have initial velocity of the reaction, and we have Vmax, maximum velocity of the reaction. Once we know the maximum velocity of the reaction, so take that point wherever the maximum velocity of the reaction is shown in the y-axis. Now take half of that maximum velocity, like half of 50 here, the maximum velocity is 50, half of 50 is 25, right? And take that point in the y-axis and bring that line, that point to the reaction velocity curve, and then drop that point from the reaction velocity curve down to the x-axis. Okay, and x-axis, as you know, it is denoting substrate concentration. Now the point is falling on number 2, which is the 2 millimoles. So 2 millimoles is the substrate concentration at which your enzyme reaches half of its maximum velocity. Because maximum velocity is 50, 25 is half Vmax, okay, and what should be the substrate concentration at which your enzyme reaches half of its maximum velocity? It is the number 2 millimoles of substrate concentration. That means at 2 millimoles of substrate concentration, enzyme, means in this particular example, enzyme reaches half of its maximum velocity. That means 50% of the active sites of your enzymes are occupied there. That's what it means. At 2 millimoles of substrate, 50% of the available enzymes active sites are occupied. So product formation is going on there. Now see the point that is meeting on the x-axis, that is number 2, which is shown here. It is referred as Km. And what is Km? Km is the substrate concentration at which your enzyme reaches half the maximum velocity. Make sure you remember this definition. It's very important to understand what is Km. Km is the substrate concentration at which your enzyme reaches half of its maximum velocity. And what is the value for Km? What is the units for Km? Units for Km, it can be millimoles per liter, moles per liter, micromoles per liter, picomoles per liter, whatever is written in the x-axis there. Okay, so Km is simply, it is the substrate concentration. In this example, what is the substrate concentration? It is 2 millimoles is the Km at which enzyme reaches half the maximum velocity, that is 25 moles per mole per second, right? Now, in general, remember how much should be the substrate concentration that takes enzyme to maximum velocity. Now we know substrate concentration that is necessary to take enzyme into its half maximum velocity is the value of Km. So here Km equals to 2 millimoles. That means what? At 2 millimolar concentration, enzyme is going into half of its maximum velocity. It is going into half Vmax. Now the question is, at what substrate concentration enzyme goes into Vmax? Is it a simple arithmetic? Is it a simple doubling? Like if 2 millimoles is the substrate concentration at which your enzyme reaches 25 moles per liter, which is half maximum velocity, at what substrate concentration enzyme reaches 50 moles per liter per second? Is it like 2 doubles into 4, and at 4 millimoles does the enzyme reaches velocity, maximum velocity of 50? Answer is no. Why? Because it is not a simple arithmetic. So in an enzyme catalyzed reaction, you can't simply double Km and say, okay, this is the substrate concentration that will take the enzyme to full maximum velocity. It doesn't work like that. So according to the experiments, it takes Km times minimum amount of substrate concentration that is necessary for enzyme to go into Vmax or to reach Vmax is Km times. So the amount of substrate that is necessary for enzyme to reach full maximum velocity is Km times 5. Okay, that is the minimum amount of substrate that is necessary for an enzyme to go into Vmax. So you got to remember that it is Km times 5. Example here, so Km equals to 2, right? Which is taking it into half maximum velocity, that is 25 per mole per second. Now, what should be the substrate concentration at which your enzyme goes into full velocity, like Vmax? It is Km times 5 means Km is 2 in this example times 5, it is 10. So 10 millimoles. So at 10 millimolar substrate concentration, your enzyme reaches Vmax. And that's why you can see, at least in this example, so at 10 millimoles, it is reaching maximum velocity. And make sure you remember that point. So, Km is the substrate concentration at which enzyme reaches half the maximum velocity, and the substrate concentration that will take enzyme into Vmax is Km times 5. Okay, now we have certain parameters here. Substrate concentration is there. We have certain parameters, that is substrate concentration is there, Km is there, Vi initial velocity is there, and Vmax is there. So all these parameters are brought together by Michaelis and Menten and given an equation. And what is the Michaelis-Menten equation? Michaelis-Menten equation is, so Michaelis and Menten has brought all these parameters together and given an equation. So what is the Michaelis-Menten equation? It is initial velocity equals to Vmax times substrate concentration over Km plus substrate concentration. So make sure you remember this particular equation, and it is necessary that you must remember this equation because there will be questions using this particular equation. Because whenever we provide value of Vmax, value of substrate concentration, value for Km, so if the question is asked what is the initial velocity of the reaction, we can simply substitute the values and get that number. Only thing is we should memorize the equation, we should remember the equation, that is Vmax into substrate concentration over Km plus substrate concentration. Okay, so and Michaelis-Menten equation, it works only for simple enzymes. Simple enzymes having a single subunit, they will follow Michaelis-Menten equation. And allosteric enzymes having a multiple subunits, they do not follow Michaelis-Menten equation. Remember that. Michaelis-Menten equation is only for simple enzymes. Now, there is a disadvantage to this Michaelis-Menten plot. Let me explain you what is the disadvantage to the Michaelis-Menten plot. It is to reach Vmax, we need to provide a sufficient substrate concentration so that we saturate enzyme. So sometimes it becomes practically impossible to provide that much substrate concentration to saturate an enzyme, and also sometimes that substrate might be too costly to provide or to saturate an enzyme. So that's a kind of disadvantage, because unless you saturate an enzyme and reach Vmax, so we cannot know how much is the half maximum velocity here, because first we need to know the Vmax to find out half Vmax, and then we find out Km, right? So in order to find out Km using Michaelis-Menten equation, we got to take the enzyme to Vmax, and that might be difficult. So that's the disadvantage of Michaelis-Menten equation. So that disadvantage is taken care by other scientists called Lineweaver and Burk. So a Lineweaver-Burk equation is the simple reciprocal of Michaelis-Menten equation, where initial velocity has become 1 over initial velocity, and the Vmax times substrate concentration over Km plus substrate concentration is reversed. It means basically reciprocal of that is Km over Vmax into substrate concentration plus 1 over Vmax. Okay, how to remember that? It will be like this: Michaelis-Menten equation equals to Vi equals to Vmax times substrate concentration over Km plus substrate concentration. So now you do the reciprocal to get my Lineweaver-Burk equation. So 1 over Vi is the reciprocal there, and Vmax over substrate has to come down, and Km plus substrate will go up. So that will be Km over Vmax times substrate concentration plus substrate over Vmax times substrate concentration. Now you can cancel out things here like substrate concentration cancelled out, and the equation that is remaining will be 1 over Vi equals to Km over Vmax into substrate concentration plus 1 over Vmax, and this is the Lineweaver-Burk equation, which is a simple reciprocal of Michaelis-Menten equation. Then what is the advantage of Lineweaver-Burk equation? Advantage of Lineweaver-Burk equation is, in order to find out the maximum velocity, so it is just a straight line that is coming up in the Lineweaver-Burk equation, where in the x-axis we have 1 over substrate concentration, in the y-axis we have 1 over Vi, or the initial velocity. So to find out that the line velocity curve, all you need is a few points so that we can draw a straight line. In the initial substrate concentration, so if we get like 2 to 3 points, like I'm in the initial velocity, in the initial substrate concentration, so if I get 3 points like this, right, and we can draw a straight line, a line is drawn straight to these 3 points, uniting that, and it will join on the y-axis, that will be a point. And then if you extend that particular point onto the other side of the 0, which is denoted as negative numbers, just to note that it is on the other side of the 0, so minus 0.5, minus 1.2 and something like that. So it is going to meet on some point on the x-axis. Now let's see what these points indicates. So the point that is meeting on the y-axis is 1 over Vmax, and the point that is meeting on the x-axis is minus 1 over Km. Okay, these are the things that you must be careful about in Lineweaver-Burk equation. So in the Lineweaver-Burk equation, it is the reciprocal indication there. So all the things that are denoted in the Lineweaver-Burk equation is the reciprocal thing. That means in the x-axis it is 1 over substrate concentration, in the y-axis it is 1 over Vi, and the point that is the line, velocity line, Lineweaver-Burk, that line, reaction velocity line, which is meeting on the y-axis, and that particular point is 1 over Vmax, and the line that is crossing over and meeting on the x-axis on the other side of the 0 is minus 1 over Km. Once you know 1 over Vmax value, So you can calculate what is Vmax. So here in this example, 1 over Vmax is 0.02. So let's write it as 0.02. Now what is the Vmax? Vmax equals to 1 over 0.02. How to simplify that? 1 over 0.02 will be 100 over 2. 100 over 2 will be 50. That means Vmax equals to 50. Okay. Maximum velocity is 50 there. So we have found out 50 is the maximum velocity using only 2-3 dots and drawn a straight line. So basically at low substrate concentration itself, we can get that Lineweaver-Burk plot and then find out Vmax. Only thing is we need to be careful that the point that meets on the y-axis is 1 over Vmax, not a Vmax. We have to find out Vmax using that 1 over Vmax. Now let's see how to find out Km using Lineweaver-Burk plot. So example here is minus 1 over Km. Always make sure you write down that minus 1 over Km equals to minus 0.5. That's where it is meeting on the other side of the 0. Now this minus 1 over Km is equals to minus 0.5. So minus minus cancelled out. You just cancel out minus minus there. And now 1 over Km equals to 0.5. So Km equals to 1 over 0.5. And 1 over 0.5 will be 10 over 5. And 10 over 5 will be 2. So that means Km equals to 2. And this is what happened now. So the advantage of Lineweaver-Burk plot is we can work at low substrate concentration and find out the Vmax instead of in the Michaelis-Menten equation. In order to find the Vmax, we have to saturate the enzyme with so much substrate that has to be provided, and that can be taken care in Lineweaver-Burk plot. And in the exams or like in competitive, non-competitive inhibitions that we are going to see later, so most of the time Lineweaver-Burk plot is used. That's why you need to know thoroughly about how to read Lineweaver-Burk equation, Lineweaver-Burk plot. Okay. So y-axis, wherever the point meets on the y-axis, it is 1 over Vmax, not just Vmax. And the point that meets on the x-axis is minus 1 over Km, not just Km. Okay. Using that, you need to find out Vmax and Km. It's a simple math that we need to do. Now let's understand what is the significance of Km. Km, as I already explained to you, it is the substrate concentration at which enzyme reaches half of its maximum velocity. And every enzyme has its own Km. It is a signature of an enzyme. And Km is independent of enzyme concentration. It explains, also Km explains, it affinity of an enzyme for the substrate. If an enzyme has got lower Km, that simply means that enzyme has got higher affinity for the substrate. Affinity in the sense that it is not the strength of bonding or strength of binding, it is at what substrate concentration that enzyme can work. So lower Km enzymes can work at lower substrate concentration because they simply have higher affinity for the substrate. Whereas enzyme that has got higher Km, they have lower affinity for the substrate. That means they need higher substrate concentration to bind with an enzyme and to make product. The best example to understand significance of Km in terms of high Km and low Km affinity feature, it is the hexokinase and glucokinase. Look at hexokinase. Hexokinase Km for glucose is 0.05 millimoles, whereas for glucokinase Km is 5 millimoles. What is the difference? So hexokinase having a Km of 0.05 millimoles, glucokinase having a Km of 5 millimoles, it is the 100 times difference. Difference is 100 times. That means glucokinase, it needs 100 times more glucose concentration to reach its half maximum velocity, because these are Km values. Km for glucokinase is 5 millimoles. For glucokinase, Km is 5 millimoles. That means at 5 millimoles of glucose, it reaches half maximum velocity. That's what it means. Km is, it is the substrate concentration at which enzyme reaches half the maximum velocity. At 5 millimoles of glucose, glucokinase reaches its half maximum velocity. And similarly for hexokinase Km is 0.05 millimoles. At 0.05 millimoles, it reaches half maximum velocity. That is for hexokinase. So that means hexokinase has got low Km. It means it has got higher affinity for glucose, and that means hexokinase work at low substrate concentration. Whereas for the same glucokinase, it needs 100 times more glucose concentration than hexokinase, because it needs 5 millimoles instead of 0.05 millimoles to reach its half maximum velocity. Okay. Now, since we know the Km for hexokinase or glucokinase to reach half maximum velocity, we can also know how much is the substrate concentration that is necessary for hexokinase or glucokinase to reach Vmax. So, what is that? If you know the Km for hexokinase, which is 0.05, or glucokinase is 5 millimoles. Let's take an example of glucokinase, which is 5 millimoles. Km for glucokinase is 5 millimoles. So what is the substrate concentration that is necessary for glucokinase to reach Vmax? So what is the answer? It is Km times 5. That means here Km is for glucokinase is 5 millimoles and times 5, that is 25. So at 25 millimoles of glucose, your glucokinase reaches Vmax. Okay, and that's how we can use the Km value to know what is the affinity of an enzyme for the substrate, and also if you know the Km value, you know how much is the substrate that is necessary to reach half the maximum velocity, and also how much is the substrate that is required to reach maximum velocity. That's the significance of Km. Now, let's look at order of reaction. So, whenever a substrate concentration is much higher than the Km, because you know the Km here, and if at high concentration of substrate, when substrate concentration is much higher than the Km, the velocity of the reaction is zero order, that is, it is constant and independent of substrate concentration. So, in my previous explanation, the third group in the effect of substrate concentration. So, it has been explained that whenever the substrate concentration is much higher, where all the enzymes active sites are full, so it has reached Vmax. So, that's the zero order kinetics or saturation kinetics. At low substrate concentration, when the substrate concentration is much less than the Km, means on the left side of the Km. So at low substrate concentration, velocity of the reaction is directly proportional to the substrate concentration, and that is referred as first order kinetics. And majority of our enzymes in the body, they are in first order kinetics. Remember that point. So in this slide, I have given what is first order kinetics and what is zero order kinetics. So when the substrate concentration is much less than the Km, velocity of the reaction is approximately proportional to the substrate concentration. And in the zero order kinetics, when the substrate concentration is much greater than the Km, the velocity is constant and equal to Vmax, and the rate of reaction is independent of substrate concentration. And the rate of reaction is directly proportional to the enzyme concentration at all substrate concentration. Let's understand this terminology here. Rate of reaction is directly proportional to the enzyme concentration. Enzyme, it all depends on how much enzyme we have. When we provide substrate, it depends on how much enzyme is present, because enzyme is the machine where enzyme is using the substrate and converting product. So when the enzyme concentration is continuously increasing along with the substrate concentration, so always you will get that proportional velocity. So here is the substrate concentration, velocity, initial velocity of the reaction, and as you increase substrate enzymes along with the substrate, so you are going to get a straight line, because we just need to add enzyme continuously, enzyme plus substrate. If both the things are added continuously, substrate along with that enzyme is also added, then you get a straight line. Get a straight line like this. Okay. And that's what is the meaning. Rate of reaction is directly proportional to the enzyme concentration at all substrate concentration. Now let's look at effect of temperature. Now every enzyme has got its optimum temperature, but human enzymes, optimum temperature for human enzymes is 37 degrees centigrade. So on either side of this 37 degrees centigrade, so enzyme velocity can go down. Okay, so if it is a hypothermia, enzyme velocity decreases. It is hyperthermia, enzyme velocity decreases at certain temperature, because as we go on increasing the temperature, it can denature an enzyme. So optimum temperature for human enzymes is 37 degrees centigrade, and we are going to maintain that. So because we are isothermic animals, isothermic means we maintain a constant body temperature, and that's how we have been designed, and the 37 degrees centigrade is maintained by our metabolic reactions and most importantly the electron transport chain that is running in our body. Now the increase in the temperature increases the rate of both non-enzymatic and enzymatic reactions, and we are going to get that bell-shaped curve where the optimum temperature for most of the human enzyme is around 37 degrees centigrade. Of course, the range can be 35 to 40 degrees centigrade. So human enzymes start to denature whenever the temperature goes beyond 40 degrees centigrade. And also when we increase the temperature, say coming from 30 degree to 40 degree, or say from 20 degree to 30 degree, 30 degree to 40 degree. So rate of most biological processes, typically they will double for every 10 degree rise in the temperature. And that's what is referred as Q10 equals to 2. Now let's move on to see what is the effect of pH on enzyme activity. So different enzymes, they have got their optimum pH, because their 3D structure, like three-dimensional structure of these proteins can vary, and the kind of amino acids that are present in the active site of these enzymes can vary. So depending on that, their pH optimum can vary, because there is a relationship between pH and pKa. Whenever pH is less than pKa, so the groups which are present in amino acids will undergo protonation. Whenever pH is more than pKa, the groups present in the amino acids can undergo deprotonation. So that kind of protonation and deprotonation will be going on, and that will alter the enzyme's action. How the enzyme catalyzes, that kind of things can be altered based on the changes in the pH. So that's what is the effect of pH here, because the ionization status of enzymes can change. As you see here in the figure, pepsin, which is active in acidic pH, so it is pH optimum is around 2, whereas salivary alpha amylase pH optimum is around 6. Alkaline phosphatase, as the name says, pH optimum is around 9. So like this, different enzymes, they have their own different pH needs, and that is according to what is their ionization status in the active site of the certain critical amino acids present there. So the change in the pH alters the ionization state of functional groups that are present in the active site, and hence they will affect the rate of reaction. And each enzyme has its own optimum pH and it functions accordingly. So extremes of pH can lead to denaturation of an enzyme protein, and usually intracellular enzymes exhibit optimal pH at values between 5 to 9. And the relationship of enzyme activity to hydrogen ion concentration, it will give rise to a bell-shaped curve that we have seen in the previous slide. Now, what are the other factors that affect enzyme activity? Enzyme concentration itself, because enzyme is the main thing here. Without enzyme, there is no product formation. So, if enzyme concentration is less and the product concentration is more, so obviously enzyme gets saturated. Or if you go on increasing the enzyme concentration along with the product substrate concentration, you can keep that reaction velocity curve straight line. So that's how enzyme concentration itself, like deficiency of enzyme, can decrease the product formation. Excess enzyme can increase the product formation, like that. Coming to the product concentration, whenever product accumulates, majority of time the end product will have a negative effect on the one of the rate determining enzyme in the pathway. So that's a kind of feedback effect will be there if there is excess product being accumulated. Presence of activators and inhibitors. Activators, as the name says, like it is going to improve the enzyme activity, it is going to optimize the enzyme activity, whereas inhibitors work negatively on the enzyme and try to decrease or inhibit enzyme activity. Availability of coenzymes and co-substrates and cofactors. So all these molecules, they will enhance enzyme activity. Optimum activity of the enzymes can be reached when sufficient coenzymes, cofactors are present. So these are other factors that affect enzyme activity. So that's about the factors that affect enzyme activity. So in this particular chapter, so we have seen the effect of substrate concentration mainly, where we have seen Michaelis-Menten equation, Lineweaver-Burk plot, simple enzymes follow Michaelis-Menten equation. We can use the Michaelis-Menten equation to calculate initial velocity, and then we have seen how, so what is the Km, what is the Vmax, and all the understanding of the Km, what is the importance of Km, and then we have seen what is the effect of temperature, what is the effect of pH on enzyme activity. That's all about this particular lecture. Thank you so much.